All right, histology. So we're going to work on doing a little bit more with the different kidney tubules. You're going to learn to kind of differentiate some of the different parts of the nephron on this one. Again, then we'll go to a slide of the kidneys and we'll take a look at each of those. Again, these tubules are part of the overall nephron. We started talking about the corpuscle last time. That is the very beginning of this. The next part of it after that is that proximal convoluted tubule. We then will go on this one part that dips down into the medulla a little bit, or a lot of it, depending on which type, which is called the loop of Henle. There's a couple different parts to that. And then we look at the distal convoluted tubule. You're going to see you're able to differentiate at least proximal from distal convoluted tubule, and then also the loop of Henle. So this is showing you that nephron. You can see the renal corpuscle. This would be the proximal convoluted tubule, goes to that loop of Henle, back up, then you have the distal convoluted tubule to a collecting duct. Those are some of those different parts that we're going to be looking at today. So the proximal convoluted tubule is what's right after the glomerulus and that corpuscle. One of the things we're going to see on this one is it is a tubule that has a lot of microvilli present on it. We're doing a lot of reabsorption here, and that is one of the things that makes it definitely distinct from the distal convoluted tubule. So proximal convoluted tubules, they're right after the Bowman's capsule. Sometimes, I think on the one image on the actual slideshow, you can actually see the start of it on that one. These ones have a lot of mitochondria. They're doing a lot of pumping, a lot of active moving of substances back out of this filtrate. Because of that, they are very eosinophilic, so more of that pink staining. The other thing is they have a brush border with a lot of microvilli, so they tend to have a lumen that looks more, like it says, they're more fuzzy but more discontinuous. You don't see this perfect cuboidal cell. You see kind of the bottom of the cell and like a little messiness on the other end. And again, doing a lot of reabsorption of water and nutrients, salts, different other metabolites that we want to keep. So if you're looking at one here, you can see it right here. You can see some of them will have a centralized nuclei in there depending on the slice. But the main thing you can see on these ones, very pink staining cytoplasm due to all those mitochondria. And the other thing is kind of a messy surface right here, where if you compare it to like the distal convoluted tubule that we'll talk about in a little bit right here, you can see those are more square defined cells. The proximal convoluted tubules tend to have a messier lumen side on that one. Very pink staining, messy lumen side. And again, you can see it here on the straight tubule versus just one of the circles there. But again, you can see kind of a messiness in the center, definitely a lot of pink stain to it. And again, the messiness is because of those microvilli. And again, just to remind everybody, microvilli are really there to aid in that absorption. They give it kind of a fuzzy look at the surface, but the whole idea is to increase the surface area, which, again, more surface area means more transport, more ability to absorb. Like it says here also, proximal tubular cells, lots of mitochondria like we were already saying, and the plasma membrane has a lot of invaginations going into this. Again, that mitochondria, I've kind of said, is a lot about the fact that we're pumping a lot of these sodium ions, among other things, through these cells in order to reabsorb a lot of that. The loop of Henle is this part that is going to dive down into the medulla a little bit, and a juxtamedullary nephron, it dives way down in. On a cortical nephron, not quite as much, but by doing something that we're not going to get into here, which is called the countercurrent multiplier, we are able to get a lot more salt and water out of these loop of Henle. They have different permeabilities on the ascending and descending loops, and it's one of the ways we can really concentrate the urine a little bit more and also make sure we're getting a lot of this water and salt back into the bloodstream with that vasa recta that it's talking about here. So this would be the loop of Henle, this part right here where it dives down. You can see there's thin and thick segments. Big difference on these ones is there's different permeabilities. This is permeable to water and not to salt. We actively pump salt out on this portion going back up. As I was already kind of saying, we have two different types of nephrons. We have ones that are called juxtamedullary nephrons, and juxta just means next to. So these are the ones that are close to the medulla, and their loop of Henle is much longer, diving way further in there. Lets us be able to really concentrate our urine quite a bit. The cortical nephrons, much shorter loops of henle, just the tip of them dipping down in the medulla. Again, they're going to look somewhat similar under the microscope, but that's what's going on with the two different types. And you can kind of see it right here. This one would be that juxtamedullary. You can see it's a loop of henle going much further down in, where these cortical nephrons is just a little bit into into the medulla there. So that proximal tubule, I mean, if you're actually able to see the junction from the side view, it goes from those cells to a completely different type. The brush border ends and it goes to simple squamous epithelium. We're going to see these are a lot of the loop of henle surrounded by these capillaries. And you can see that thin portion is what it's talking about there. That's going to be that simple squamous. And again, you can see simple squamous going from cuboidal to simple squamous right there. If we look at these under the microscope, you can see these small little circles here with single cell walls of simple squamous with lots of blood vessels surrounding them. Those would be those thin parts of the loop of henle. Probably the one part of the loop of henle I might make you take a look at and be able to identify, but you can see there's another one right up there. Again, as it goes back up, you're going to get another very abrupt change where it's going to go to these distal tubules. Again, no brush border on these ones. They're going to go back to cuboidal cells, especially as it gets into this cortex again. We're going to get a, it will go near that the renal corpuscle. It's called the juxtaglomerular apparatus. It does a lot of, simple sense, a lot of regulation in terms of urine formation and monitoring what's in that urine, making sure that the filtration rates are good. Again, something I don't want to go into in histology because this is a much more of an A&P topic in terms of the physiology of that. But you can see this would be that thick ascending limb right here going to that distal convoluted tubule. And again, you can kind of see it thicken up here, and then you go to these ones right here, and again, into these ones right here, and again, you can see it does not have those microvilli present on it. If you're looking at these ones, these straight tubules, again, we don't see that messiness on the interior here. They tend to be much cleaner looking. You can also see this macula densa right here with this glomerulus. This would be a piece of distal convoluted tubule right here. That group of cells in the middle is called that macula densa. That's where that juxtaglomerular apparatus is. Kind of have to get a good view on these ones. We'll see if we can find one when we're doing one of the digital slides. But that is, again, what is monitoring what's in that distal convoluted tubule and making changes to the filtration rate by messing with the efferent and afferent arteriole. And it's kind of showing you, here's the afferent arteriole, here's the efferent, and you can see these cells are kind of in between those ones with a piece of distal convoluted tubule right here, where you can see how they can be highly involved in kind of monitoring that and then influencing that filtration based on the afferent and efferent arterioles. So that distal convoluted tubule, it looks a lot like that distal straight tubule of the loop of henle. Again, main thing on this one, a little bit of secretion taking place there. Depending on if there's certain signals present, it may reabsorb some more water. So if you're releasing an antidiuretic hormone from the pituitary gland, it will reabsorb some water there. Aldosterone is also going to modify the ability of these tubules to reabsorb more sodium if need be, which tends to make water follow that because water tends to follow salt. So you can see the distal convoluted tubule on the image right here lit up. And again, you can see it on this one right here. Again, if we're looking at it under the microscope here, you can see the big difference. Eosinophilic cells, these would all be proximal convoluted tubule. You can see how that distal convoluted tubule, much cleaner, lumen, looks like a standard cuboidal cell. And the other thing we don't see is we don't see that intense pink cytoplasm. You tend to see the nuclei a little bit more, and the cytoplasm tends to be a lot lighter in color. So you can see here and this one, and probably a little bit more of them right along here. Those would all be distal convoluted tubules. The collecting duct this is after multiple nephrons. They're going to dump into this tube that's going to go down and be sooner or later to the renal papilla and to the minor calyx. These are lined by simple cuboidal epithelium, but they kind of start to look a little bit bubbly. So they get a little apical membrane bulge. And these are the ones that, again, under the right signal with the antidiuretic hormone, they can reabsorb more water. You get more of these aquaporin channels going to the surface and can help pull more water out if need be. If, let's say you're not bringing in enough water, or you're in really hot conditions and you're sweating a ton, we might release a lot of ADH in order to get more water staying in the body. But this is what I was kind of saying with those collecting ducts. You can see how they start looking a little bit. So here's the lumen. See how they have this luminal bulge? Like I said, they look a little bit bubbly. And that's the big differentiating characteristic of collecting ducts. So what we'll do is we'll look at these on the side here, and then the last show of this one is really just going to be taking a look at the other parts of this, which would be the ureters, the bladder, and the urethra. So what I'm going to do here is got about 4 different digital slides we're going to take a look at here real quick to try to show you each of the different things. I've already kind of zoomed them in on the areas. Usually I'd like to try to kind of show you where I'm zooming into, but on this one, because each of these are kind of distinct areas and I want to make sure I can find stuff and not waste a minute looking around for the right stuff and killing time with you guys. This one, if you look over at the screen right here, the box is actually in this area right here, so you can kind of see if this was the cortex and more of the medulla, we are up in the cortex. So what we're looking at here, you can see a number of those glomerular capsules. There's one here and one over here. And then what we see is a lot of tubules surrounding that. You can see all these ones right here. See how they're nice dark pink staining? Don't have a particularly clean interior on these ones. All these ones right here would be considered proximal convoluted tubules. As we look in close proximity to those, you can see some of these tubules which have a much less kind of pinkish to them, more obvious nuclei, and a much cleaner luminal side. This would be a distal convoluted tubule. These are distal convoluted tubules. If you look around a little bit more, you can generally find even more than that. Here's probably another distal convoluted tubule right there. So those show you kind of the differences between the proximal and distal convoluted tubules. I can see if I can find one more real quick example of it right here. Again, proximal convoluted tubules here and here. Distal convoluted tubule right along there. You can tell the difference in what the cytoplasm looks like and presence of nuclei and cleanliness of that luminal surface. So what I'm going to jump to next is one that showed you just the specific glomerulus and kind of that one pole on it. So this is a glomerulus right here. This would be that little distal convoluted tubule that goes up in between the afferent and efferent arterioles, and the macula densa would be these cells kind of in between those. So that's what I just wanted to show you on the one there. These aren't the easiest things to find. I don't know if I'll go there on a test on this one unless I can find some good examples of it, but that shows you that piece of distal convoluted tubule that's going up in between. So the next slide I was going to show you is just a hunk of the cortex on this one. We are over in this region right here, but the nice thing about this region is you can see a lot of these cells right here. All these that we're zooming in on here, these are mainly different parts of those loop of henle. So you can see the much skinnier cells, the skinnier lumen, so some of the straight tubule, probably right here, and then as we find some that are a little bit thinner, you can see as we look at some of these, some of that simple squamous in these areas where there's a much thinner tubule on those ones. The last thing to kind of show you is we'll go back to this slide right here. So we're down now. Here would be the cortex. Here's the medulla. We're down deep in the medulla in this region right here. If you look at a lot of these tubules right here, these are those little bubbly tubules that we were talking about. So as you look at those two right here, you can see kind of those bubbled luminal surfaces. Those would be those collecting ducts. Again, pretty clean epithelium on that one with a little bit of a bulge on the surface on those ones. So collecting ducts here, another one right down here. Again, as you're looking at these kidneys, if you have a slide that's like this, most of your tubule types are going to be up in the cortex here along with your glomeruli. As you are looking for kind of loop of henle and collecting ducts, you're going to want to go down more into the medulla of these structures here.